So there are a lot of really good reasons that we would want to isolate DNA from organisms. A lot of it has to do with just basic study of organisms. We want to know what their DNA is. We can look at how closely related we are based on DNA. But there's a lot of practical applications of this too. So, let's say we have this crop field and we have crops that are growing, but we're trying to grow them in the desert Southwest, and we're having lots of drought, and these plants are not surviving very well. But we notice that there's some weeds around that are surviving really well despite the drought conditions. So we can actually isolate that drought resistance gene from these weeds, and then we can splice it in to our crops so that we can have crops that are more drought resistant. So that's one application. Gene therapy is a classic one that you've probably heard a little bit about, but maybe not know what that is. But in gene therapy, we might have somebody who has a mutation in their DNA that we want to fix. And so we have to get DNA from some healthy person. So we'd like swab somebody's cheek that's healthy. And we want to be able to isolate that DNA and pull out that sequence so that we can put it in a virus and get it into the cells of the sick person and actually change their DNA. So that's another really great application. And we're going to learn more about that later. Vaccines. Vaccines are a huge one. Every year, I don't know if you've noticed, but every year you have to get your flu shot again, and that's because the flu virus evolves every year. And so what scientists do in creating vaccines is they pull the DNA out of a virus and out of the neighboring viruses, and we compare them to see how closely related they are. And we build like these phylogenetic trees to see who's related to who, so we can track the direction that these viruses are going and try to predict the next flu virus. Or if we're trying to figure out like who infected, where was the origin of the infection, we can take a look at the patients and we can do the phylogenetics and we can trace it back to a single origin and try to figure out where it originated and if there's something we need to change in human behavior to stop getting infected with these viruses. So there's a lot of practical applications of this. There's lots more that I haven't talked about, but isolating DNA from a cell is a really important biotechnology technique that we use.